Hello, everyone. I'm Julia. I'm now a professor at PUCRS University in Brazil. When I submitted this research, I was a PhD student, but I already got my PhD degree. So today we'll present this paper entitled Automatized Data Integration in Hadoop-based Data Lakes, which is the main subject of my PhD thesis. So let us start by talking about the background and what motivated us to develop this work. Regarding the background, the big area is big data, where a data lake is a technology for managing big data. And Hadoop is the most used tool for building data lakes. And we used data profiling techniques to understand more broadly what are the characteristics of the data that are in the data lake to facilitate data integration. And now let's explore a little more these terms that are part of our research. So big data are data that have such a high volume, velocity, and variety that we need to use specific technology to transform this data into valuable insights. But if we have so many information assets which vary in size, type, volume, where we are going to store all of this data at runtime so that we can access this data whenever we want, right? This is where the data lakes comes in. And we developed a previous study where we defined the data lake as a central repository system for storage, processing, and analysis of raw data, in which the data is kept in its original format to be processed only when we need. And data lakes can store a varied amount of formats in big data ecosystems, from unstructured to semi-structured and even structured data sources. And the most used solution for creating data lakes nowadays is Apache Hadoop, which is built based on the Google file system idea. Hadoop is a framework that makes it possible to create clusters with commodity hardware for managing big data. And it helps us with the scalability issue. And data integration, it allows us to combine different data sources to provide the user with a unified view. And there are some different approaches for data integration, such as the one based on set similarity, where we can use the intersection among the sets to find candidates for data integration based on the distinct values. And we decided to perform experiments with datasets from the bioinformatics domain. We know that there are several different data sets in bioinformatics, and sometimes they have to be manually integrated, which is subject to error, and it's also time costly. So our proposal here is to use data integration to use data profiling techniques to facilitate data integration. And it's all based on a Hadoop data lake ecosystem. So let us now present our problem statement. Managing and querying a data lake is not an easy task. It's a difficult task, mainly because the data is heterogeneous. The data can have another problem, such as it can be duplicated, it has considerable volume, and also sometimes the data have quality issues. Additionally, big data integration tasks are dependent on experts, and it's error-prone and time-consuming, as we stated before. And regarding the complexity of the problem, this is the equation to calculate the total number of comparisons that need to be performed to find the columns candidate to data integration. D is the number of data sets, in our case, 9 data frames. We worked with 9 data frames. And M is the average number of attributes for each data set. Considering the data sets we selected to perform the experiments, in our case, we would have to perform about 52,000 comparisons among the attributes to identify the candidates for data integration. So it brings us to our research question, which is how to create a model to automatize data integration for heterogeneous data sets, taking advantage of the Hadoop-based data lake ecosystem. Then let us present how we approach the research problem and related question. Let's start by our hardware and software infrastructure. So our data lake is supported by a Linux server with the Hadoop framework, and we developed our model in Python programming language in this hardware infrastructure. Regarding the composition of our data lake, it is composed by Apache 95 for data ingestion, Hadoop distributed file system for data storage, Python for data processing and integration, and Neo4j we use for data presentation to present our results. And to perform the experiments, we selected these 8 well-known datasets from the bioinformatics domain. We selected pharmaceutical datasets such as Drug Bank and Drug Central, other datasets about genes such as Oming and DisGenet, also protein-related datasets such as UniProt, Reactomy, and IID, and an ontology for disease definition named Mondo. This table shows that we selected heterogeneous data sets because they have varied sizes from 1 megabyte to almost 2 gigabytes. We also have different amounts of instances or entries, and with the number of attributes varying from 6 to more than 2,500. We also present different formats such as text, CSV, XML, JSON, and we also query UniProt using API. Thus, those datasets present a variety of format, size, and number of entries, which are very important so we can validate our approach. And what we did with those datasets was to check for similarity among the attributes in the datasets. And then we suggest which ones are more similar and therefore should be considered as having the same information, the same set of information. So we could suggest that these columns are candidates for data integration. And we start the experiments with manual integration, a manual analysis performed by a domain specialist. The specialist analyzed all the data sets and a sample of the data and the name of the attributes to perform this manual data integration. In this figure, the nodes present the data frames names and the edges are the attribute names. The directions of the edges indicates that the attribute is an integration point. For example, the attribute label from Mondo, it is also contained in one of the attributes of the DisGenet dataset. So to start data ingestion, we created processors in Apache 95 to get the data from the sources. We created one processor for each data set, and then we get it from the sources, web sources, usually. And we save these data sets into Hadoop HDFS to be later processed using Python. And now I'll start explaining about our data processing and integration algorithm. We defined some restrictions regarding the attributes. Some are configurable, others are not configurable. Among the configurable restrictions, we can define a number for the amount of unique values that the attribute must have to enter in the comparison. We can also configure if we accept comparisons among binary, null, or numeric attributes. Regarding the non-configurable restrictions, our algorithm do not perform redundant comparisons among data frames, nor comparisons among attributes, or among the attributes that belong to the same data frame. The algorithm starts by creating data frame from all the data sets we ingested into our data lake. Then the next step is selecting a source data frame, which will be the one to be compared with the other data frames. Next, we select the first attribute to be compared, then the first candidate data frame, and then the attribute on the candidate data frame. So finally, we check the similarity between the two attributes, and if the similarity is above a predefined threshold, we print the similarity or we add an edge among the data frame nodes on Neo4j. And we repeat this step until there is no more data frames or attributes to be compared. Regarding the threshold, it represents the minimum size of the intersection between two attributes. So we can consider those attributes as candidates for data integration. The threshold is configurable, and in our case, we defined the value of 40% after testing in our data frames to avoid having unlinked nodes. Sorry about the noise, I think. I don't know if you are hearing, but my neighbors are having a party, so if the noise is too high, I maybe play my video. What do you prefer? I don't know if you are hearing the noise, but for me it's too high.
I mean, if you're more comfortable that way, that's fine. But we can hear you. I think I can hear you anyway. But if you would rather play the video, it's fine as well.
Okay, I will keep it. And if the noise is too high, please let me know.
Thank you.
The processing phase, it is based in an in-memory set similarity approach because we use Python notebooks. So we use some of the most well-known distance metrics for set similarities according to a previous systematic literature review we performed. So we used similarity metrics, the overlap set similarity, the Jaccard index, the Sørensen-Dice, and Tversky coefficients. The output of our algorithm is a list having the names of the data frames, attributes, and resulting values for the similarity metrics. In this case, when the resulting value is equal 0, it means that the attributes are not at all similar. While if the similarity value is equal 1, it indicates that the attributes contain the same data. And here we present our final data integration in Neo4j with all the points of integration identified by our model. Here we have a table with the same information of the previous figure, but here we also have more details about the overlap and Jaccard indexes. And we also indicate the integration points which were manually mapped and the ones only identified by our model. So the overlap coefficients represent better how similar two attributes are and the level of data integration that we could achieve if we integrate two data frames in our experiment, at least. It provided us better insights about how to solve data integration tasks. For example, the attribute drug bank ID, which is presented on both drug bank and drug bank protein, it represents the same data according to the overlap coefficient and according to the manual analysis. But in this case, the Jaccard index is less than 60%. The manual mapping performed by specialists found 10 integration points, while our solution identified 32 integration points, including 9 of the manually integrated. And for one of the manually integrated points, we suggested 2 better integration points than the one mapped by the specialist. So we discovered 22 more paths of integration that were not manually identified, such as from UniProt data frame to OMIN and DisGenet data frames. Now let's head us towards the end by explaining a use case where our model could help a data scientist who wants to study the tuberculosis diseases having the gene name. If we Google the gene name, UniProt, which is one of the data sources we used, may be on the top 5 results. It is easily findable in UniProt. And UniProt is easily integrated with OMIN, DisGenet, IID, and Drug Central using the attribute gene name. With the integration with OMIN, we would find information about genetic phenotypes, while integrating with DisGenet would bring the variants of the genes related to the tuberculosis disease. IID would bring information about how a protein related to the gene interacts with other proteins. And Drug Central would add information about how the drugs interact with tuberculosis. The integration with Drug Central will allow us to integrate with Drug Bank, which brings details on the substance of the drugs and related products. For instance, what medications could be used for tuberculosis treatment? And having UniProt data, we can also integrate with Reactome to understand how the molecules interact in a cell to change the cell or to create a specific product. Finally, we could connect DisGenet data with the Mondo ontology and learn about the different types of the tuberculosis disease, such as ocular and spinal tuberculosis. To conclude our presentation, for future work, we plan to implement text similarity strategies to magnify the reach of our results and also to increase the points for data integration based on textual semantics and synthetic. And we want to acknowledge that this study was financed by CAPES Brazil, and we also thank the Newcastle University for the support and advice in the early stages of our work. That's it. Thank you for your time.